Thank you,
Take it
Lindsay.
away, Haviv.
Thank you, Lindsay. And thank you, everyone, for coming here. Wow, it is a packed room. So let's dive right in because we've got a lot to cover. And this might go a little bit over 45 minutes. So, okay. Cool. So before I dive into my research, for those who maybe don't know me in this room, I'll just share a brief background on who I am. So my name is Haviv Avrahami, right? And like Lindsay said, I've been here in the department for a while, also working at the North Carolina Museum of Natural Sciences in the paleontology lab. Okay, so believe it or not, this slide right here is probably the most text-heavy slide in the talk, so don't worry too much. But just a brief outline of what we're going to cover is there's 4 chapters. Chapter 1 and chapter 2 are split up into 2 parts. And we're going to be starting off with the published osteology that came out last year, focusing part 1 on the background of this animal and its context in space and time. Part 2 is going to focus on how we build evolutionary relationships, specifically how we did it for this group of dinosaurs. Chapter 2 is going to focus on the detailed cranial osteology, and where I'm going to show some of the methods that I used to accomplish this part of the chapter. And then the part 2 of that is going to focus on the variation that we observe between multiple individuals. Chapter 3 is going to focus on assessing the growth of these animals revealed via histology. And then chapter 4 is going to be a short foray into our investigations of possible signals about fossoriality, otherwise known as burrowing behavior. So let's jump right in. Okay, so today my talk focuses entirely on this critter right here. This is the star of the show. But before we can talk about the star of the show, we have to set the stage, okay? To do that, we have to go back in time approximately 99 million years to the Cenomanian age at the very start of the late Cretaceous period, colloquially known as the mid-Cretaceous. Right now, we're zooming in here on Utah back then, and we're focusing in on that little red strip. So during this time, there was an event going on across and underneath Western North America called the severe orogeny. And it was quite severe indeed, because there was convergence that was happening between the North American tectonic plates and various oceanic plates that were coming in from the west. And this led to compression and deformation of Earth's crust, initiating a plethora of mountain building events and volcanic activity. But when one thing goes up, another must go down. And due to the subduction of the Farallon plate beneath North America, coupled with rising global sea levels, this led to a subsidence of the continent's interior seaway. And these new low elevation regions allowed for that massive intercontinental seaway to form and bisect North America longitudinally. And so it was at this terrestrial interface between the mountains in the west and the seaway in the east where a number of accommodation zones formed. And these accommodation zones are areas where all that volcaniclastic material and all that eroded mountain sediment was able to accumulate, ultimately bearing and preserving a number of prehistoric ecosystems that existed along this margin. And so that is where our story begins. At the distal back bulge of the San Rafael Swells Accommodation Zone in central Utah, this is the Mussintucket member of the Cedar Mountain Formation. Now, the Mussintucket was a warm and wet floodplain ecosystem that is very similar to the shores of modern-day French Guiana. And it was populated by a mosaic of remarkable creatures that were fighting for survival at a time of prolonged and complex environmental changes and a prolonged extinction event, conditions which closely mirror the conditions that we face in our modern day climate change and current extinctions. And so because of all the sediment input that was coming down from the mountains on all that volcanic material, the Mussintucket is predominantly composed of cat litter, otherwise known as bentonitic clays. And in our lab's experience, these mudstones somehow become both the slipperiest substance on Earth while also sticking to everything that it touches. It can be seen here with Emmy and her hobbit feet. So why is the mustn't touch it important? And in general, why is the mid-Cretaceous important? Well, over the last century of paleontological science, there's been an extensive amount of work that's been done to uncover the biodiversity of the Jurassic and the latest Cretaceous. But as you can see, the biodiversity of these two time periods is vastly different as they're separated by more time than the time that separates us from the dinosaurs. And this leaves a huge gap in our understanding of how dinosaurs evolved and diversified across this vast amount of time. And that is why the mid-Cretaceous is so important, because it preserves a record of this evolutionary transition. And over the last few decades, our lab and our lab's associates have worked extensively to reveal more about this previously unknown period in Earth's history. So, going all the way back to 2015, while I was actually still an undergrad, I went out to Utah for field work with the North Carolina team. And it was that year that Lisa found something really cool sticking out of the ground, not far from our base camp. And so as all good geologists do, we dug down to see if there was more. And to our surprise and amazement, we found the skeletons of two incredibly well-preserved dinosaurs, which would ultimately become the crux of my dissertation. But what was even more exciting is with that with every subsequent year that we went out, we continued to find more and more of these localities preserving seemingly the same type of dinosaur. This provided me a relatively large sample size of animals to work with. So here's a close-up of the mini troll locality. Those individuals on the left, what they look like underground taken with CT, and then on the right, you can see the published quarry map. And so thanks to our colleague and our co-author, Dr. Ryan Tucker, he ran a series of high-resolution uranium-lead zircon geochronological isotope analyses across 4 laterally extensive volcanic bedded ash layers. And because of that, our lab has been able to resolve some of the highest resolution terrestrial age dates during the Cretaceous, bracketing some of these localities by only into a 60,000 year window for a time period that dates back nearly 100 million years. So that's exceptional resolution. So from pretty early on in my project, I was able to figure out that these animals that we continue to find belong to a group of dinosaurs that look something like this. Now, there's not a great name for these dinosaurs. Historically, they've been referred to as things like hypsilophodontids or neornithischians, or probably most commonly, generally ornithopods. But all these names are problematic due to either being taxonomically invalid, insufficient, or just too broad. So we're calling them exactly what they are, what they're shaped like, and they are simply small-bodied, early diverging ornithischians. Here and now, I'll be abbreviating that as SPEEDOs for short. So if we go back to that orbital view of North America during the Cenomanian, you may have noticed that there's that little pink spot approximately 600 kilometers to the north of the Mustn't Touch It. And that is the Wyand-Von assemblage of Montana and Idaho, which possibly and partially overlaps temporally with the Mustn't Touch It. And there's already a speedo that's known from the WVA, and this is Eryctodromius. So one of the initial hypotheses was that, hey, perhaps these new specimens that we're finding are just new additions to Eryctodromius. However, it's important to consider something, and that's that the WVA depositional system was deposited in the 4 deep of the San Rafael swell, and therefore located further away from the interior seaway, resulting in a more arid environment that is distinct from the Mustn't Touch It, and raising the possibility that perhaps we have 2 distinct species with different habitat preferences. And indeed, similar species distributions and overlapping or near overlapping habitat preferences exist among many modern medium and small-bodied herbivores such as deer. So in order to determine if our new Mustn't Touch It speedo constitutes a new species, I had to do a series of museum collection visits around North America, but also in Europe and Asia, to collect comparative data on a plethora of relevant speedo species. And this allowed me to take these bones side by side in high resolution and map the differences. So I'm just going to show you a few examples of what some of these were. This is an example of the second spinal bone in the vertebral column. This is the axis, and in the Mustn't Touch It speedo at the top left, you can see that the angle of inclination between the dorsal surface of the neural spine is in parallel with the sutural surface to the centrum, whereas in all other speedos, this angle is inclined. Here's another example of a comparative observation that was initially thought to maybe be unique, but we later came to find out that it seems to be intraspecifically variable. But this is just an example of in the must-and-touches speedo, there are these massive intercondylar voids between vertebral number 2 and 3 that are absent in the others. Or here with a collection of shoulder blade bones, where I was also mapping some of the differences between a collection of different speedos. So altogether, these observations confirmed my hunch that the shape of the bones of the must-and-touches speedo are more similar to Oryctodromeus than any other speedo. So what does this mean? Does it mean that it is Oryctodromeus? Well, not so fast. Because if we look at some, this is a simple quantitative analysis, and this one's called a principal component analysis. We can see that the scapula of the must-and-touches speedo form a cluster that is close to but distinct from Oryctodromeus. But this alone is not enough. What we really need are something called autapomorphies, and this is arguably the most important figure from this recent publication. This on the top in the red is the autapomorphies. These are what you need to erect a new species. They are features that are unique to just that taxon, but the ones on the bottom, the synapomorphies, are also important because those are shared characteristics which allow us to align it in the evolutionary tree with our observations. So all that said and done, with all that, last year we were able to publish this new dinosaur and finally give it a name, and we named it Fona herzogae. All right. And I'll talk briefly about what the name of this is, what the name means, but dinosaurs, fossils usually get two names when you name them. There's a genus and a species name, right? And so the genus name Fona comes after the ancestral mythology of the Chamorro Islanders, which is what my people are. And so I spent some time communicating and getting in contact with a number of the indigenous leaders on the island and some other some other folks that have background in a lot of Micronesian and Polynesian studies. And we talked story for a while and landed on the story of a brother and sister spirit, right? The brother Pontan and the sister Fona. And when the brother died, the sister Fona used the parts of his body to create the pieces of the universe and the island, right? And then when she died, she became petrified within the earth, and from her body came forth new life. I really liked how this story closely mirrored the life and death of Fona the dinosaur, which was also preserved under the ground, which we'll get into later, and things like that. The second part of this name, herzogae, honors Lisa right there. So me and Lindsay decided to honor our operations manager, Lisa, who has been one of my strongest allies and confidants all the way back to 2014 when I rappelled into the paleo lab from the rafters and asked for a job. So, okay, part 2 of chapter 1. This is where we're going to talk about evolutionary relationships. So broadly speaking, there are 3 main taxonomic groups of dinosaurs. There's the mostly meat-eating saurischians, the long-necked plant-eating sauropods, and the plant-eating ornithischians. And Ornithischia is by far the most morphologically diverse group of dinosaurs, as can be seen here with the most well-known groups. And for the longest time in children's science books and posters and documentaries, this is basically how you've probably seen their family tree presented. But when we crunch the numbers and when we run our statistics, what we actually get is something a lot more like this. And in the study of evolutionary relationships, we call this the dreaded polytomy, which is not a good thing because it's not a reflection of reality. It simply means that we don't yet have the data and resolution necessary to figure out exactly who is related to whom. And this is quite a challenging undertaking to resolve because the fossil record is so incomplete and we don't have the genetic data like we do with modern animals to utilize. So just to highlight how challenging that this process is, I want you all to imagine that you're going to your friend's family reunion, and when you walk through the door, everybody except you gets amnesia. And then it becomes your job to figure out how everybody is related to each other. So if we use my family as a case study, and we zoom in up here, Would you ever believe that that guy is my cousin? This guy is technically my uncle, and this weirdo here is my stupid brother. And there's Emmy for scale. So if we go back to the family tree, the reason we have this polytomy is right there on the left. That's the quagmire. That's the quagmire of speedos, right? Okay. Now, like I said earlier on in chapter 1, I knew from my initial observations that Fona was most probably closely related to Eryctodromius. However, I needed to run something called a phylogenetic analysis to check my qualitative observations against the raw quantitative data. So I hypothesized that Fona would fit in right here next to Eryctodromius within a group of North American speedos called orodromines, right? Orodromines. This is going to be important later on. Now, these orodromines seem to have been prolific in North America throughout the Cretaceous period until right before the very end, when they were inexplicably replaced by the Thescelosaurians. Now, I know it's probably hard to visualize the nuanced distinctions between these groups, as that's a level of evolutionary granularity that might be only familiar to primary researchers. So just to visualize what these distinctions are between these two clades, we can think of them as something maybe like the differences between musk deer and elk, or maybe they're something like this, or maybe it's something like that. But that's just the kind of level of differences we're looking at. Okay, so in order to perform this phylogenetic analysis, I needed to score hundreds of bony features for Fona into a data matrix, right? This is like the world's largest Harry Potter sorting hat. And in these matrices, these include morphological information that has been discretized into binary, ordinal, or multivariable data, but not continuous data, for approximately 80 ornithischians in this case, spanning all the ornithischian clades, but focusing mainly on speedos as the high-resolution area of interest. Now, I didn't do all this by myself. The majority of these codings represent decades of previously published observations by other researchers. But what I realized pretty early on is that there were a few problems. First, there were multiple matrices that were used by different authors in the past, each focusing on different parts of the trees, but each containing relevant data for speedos. So I spent several weeks merging all these matrices together, but during this process, I discovered that there were a number of disagreements between the different authors and their observations, particularly in the validity of particular characters, as well as a number of redundant character codings for the same feature multiple times or coded in different ways. There were also a number of misinterpretations and just general logical fallacies associated with some of the fundamental constructions of a number of characters. But fortunately for me, I had collected so much 3D data during my museum visits that I was able to update a number of speedo characters across the matrix while also adding in a number of my own observations. Ultimately, I ended up with this new behemoth of a matrix composed of around 600 characters spanning the entire breadth of Ornithischia, ornithischian clades. And so I ran this new matrix through something called Bayesian and parsimony statistical analyses. Now, I won't go into too much about what the differences between these two statistical programs and approaches are, but basically parsimony is the simplest approach that seeks to find the tree that minimizes the number of evolutionary changes or steps, whereas Bayesian uses probability to find the most likely trees based on a series of models and parameters. Now, at the end of the day, here's the big reveal, okay? We're going to look at the Bayesian analysis here. So drum roll, please. We're going to zoom in on that white void area. Boom, 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 and boom. Fona and Eryctodromius are not orodromines at all. They are Thescelosaurians, and this is a pretty big deal because it means that Thescelosaurs didn't just pop up out of nowhere right at the end of the Cretaceous, but they were already a well-established faunal agent during the mid-Cretaceous. And it also means that Thescelosaurs and orodromines existed simultaneously on the North American continent during the Cretaceous, but strangely enough, we have yet to find them living together in the same paleo environment, suggesting that perhaps they have different ecological preferences, or maybe we just need to increase our sampling. Okay. To bring it all full circle and connect the qualitative anatomical observations I mentioned in part 1 with the quantitative stuff, this is where all that manifests in the quantitative realm. So this is an example of one particular character that's really strong, right? This is the jugal. This is the upper jawbone of the cranium. We're looking at the lateral surface. And in orodromines at the top, they possess this really pronounced jugal horn that projects laterally and somewhat backwards. So they in the character matrix are coded as a 2. Don't worry, there's not a difference between 1, 2, and 3 in terms of like scaling. They could be A, B, C. They're just representing states, okay? And then in the bottom, Thescelosaurians are coded as either a 0 or 1 because the lateral surface of their jugals is either completely smooth or they can be a bit ornamented, they can be rugose, and they can be somewhat swollen, like a pimple is on the surface, but they never have a prominent jugal horn emanating from the lateral surface. Now, it's not always this easy. Sometimes the characters can be more ambiguous. This is the example of the medial surface of the scapula near the neck. And in Thescelosaurs at the bottom, we're looking at Phona, Oryctodromeus, and Thescelosaurs from left to right. And Phona has this medial, the Thescelosaurians have this medial tubercle at the base of the neck that is absent in orodromines, but you can imagine that this is a pretty subtle feature, and if the light's not hitting it at the right angle, you might miss it, for example, in Thescelosaurus. Okay, that was the end of chapter 1. Now we're moving on to chapter 2, where we'll start with an overview of my cranial osteology and briefly look at the novel methods I had developed to make this chapter pop. 142 years ago, a famous British geologist named John Holck, who was a geologist and a surgeon, published an anatomical manuscript on one of the very first dinosaurs that was ever discovered. This was a speedo named Hypsilophodon. Now, back then, any natural scientist that was worth their weight in gold would have also been an exceptional artist capable of converting their visual observations into exquisitely detailed anatomical drawings. And although their full body reconstructions were inaccurate and downright silly at times, right? Like Hypsilophodon was not a lizard, it was bipedal. Their osteologies and their figures still remain incredibly useful and informative to this day. But unfortunately, in the preceding generations over the last 100 years, with the advent of photography, faster publication demands, and a general disillusion between the arts and the sciences, this valuable craft is somewhat diminished in prevalence. But fortunately, technology has come a long way, and with the power of 3D modeling, even the most artistically uninclined researchers are able to convert their research objects into exceptional resolution figures. So we go back to Phona. I'm sure you've noticed up to this point that I have yet to really discuss the skull in detail, and that's because the skull bones are quite small, averaging only a few centimeters in length, making traditional photogrammetry and commercial scanning either impossible or inadequate. Right? And so although I was able to do a lot with the postcranial bones because they're bigger, the skull bones, I was left with naught but hopelessness. Until I found something called mobile device scanning, which I like to call pocket photogrammetry. So back around 2015, our mobile phones in our pockets started to support 3D information. Initially, this was fueled by ridiculous things like selfies and stuff like that, but it's a powerful technology that has continued to increase in its power. So what happens when you take a picture with your phone? Well, every time you take a picture with your phone, you're not just capturing a flat layer of JPEG color pixels. Instead, a cacophony of computational events occurs in tandem, collecting swaths of hidden metadata that are used to reconstruct the 3D model of interest. All right, and so around 2022, I partnered with a company called Metascan that has now been rebranded as Abound, and together I worked with them to improve their application to better support the needs of not just paleontologists, but also archaeologists and museum creators of all types. All right, so this is just an example of what that interface looks like. Left, we have a tiny hadrosaur tooth about the size of a pencil eraser, and on the right, we have the full skeleton of Phona. This is a direct screen grab from an iPhone. A few years ago, I did a detailed compare and contrast on these, which I won't talk about too much, but you can see on the left is a Toyota Camry priced commercial scanner, and on the right is a $500 iPhone. And in many cases, the vast majority of cases, especially with smaller elements, pocket photogrammetry methods vastly outperform some of the commercial scanners, both in terms of texture quality and geometric fidelity. And so with these methods, ultimately, this allowed me to scan all those tiny pesky cranial bones in extremely high fidelity, allowing me to fully reconstruct the skull using a video game CGI software called Blender, where I was able to attach anchor points and hinge points and get accurate rearticulation of the skull that had previously been disarticulated. Okay. So why is all this important, and why are osteologies important in general? Well, osteology is the bedrock foundation of paleontology. You can think of them like the instruction manual for a Boeing 737, where every nut, bolt, connection, and update from previous models are specified in detail. These osteologies serve to establish taxa as discrete units of data, like the kilogram is to physics and hydrogen is to chemistry. And without these osteologies, misinterpretations can result in future studies. And I've seen this firsthand in my case, where there was a speedo that was described as having a particular feature that was misinterpreted, resulting in a supposed ghost lineage that extended a mid-Cretaceous clade all the way back 80 million years to the Jurassic, or would have, right? But this was not the fault of the authors of that. They simply lacked a medial view of a jugal bone for any speedo. So that's just one reason among many why osteologies are important. Now here's a screen grab of that upcoming publication for each of those skull bones from multiple individuals. And if we zoom in here, we can see that there are multiple exemplars of the same type of bone. And not all these bones are identical in shape or features. And that's because Fona is not represented by one individual, but around 7 individuals to date, which will probably increase even more in subsequent years. And across populations of species, variation exists, which brings us to part 2 of chapter 2, intraspecific variation. That's the question, what's up with all this variation in Fona? And to determine that, we need to go over briefly the different types of variation that exist. The first is taphonomic variation. This is simply the effects imparted upon bones after death, and can include things like crushing from other animals. It can include biological weathering, scavenging marks, bite forces, just general weathering. But it can also involve the diagenetic effects that are imparted upon buried bones after death. And this can include things like warping and squishing due to tectonic forces. There's also another type of variation called interspecific variation, which is simply the differences between different species, right? So such as these crocodile and alligator on the left, or different species of hadrosaurs. And then there is intraspecific variation. And this is the variation between different individuals within species populations. All right, but there's multiple types of intraspecific variation, which is what we really need to cover. The first is ontogenetic variation, and this is the differences between individuals at different life stages, right? Babies look different from adults. If you took a baby and you scaled it up to the size of a human, it would look horrendous. It would have a disproportionately gigantic head for the rest of his body. Right? This is the same thing that happens in animals and speedos. In this case, one example is that over ontogeny, their skull elongates, they can grow additional teeth, things like that. There's another type of variation, and this is sexual dimorphism, which is the differences between sexes. Sometimes these can be pronounced skeletally, such as in many modern mammals, like those with horns and those without. But they can also be more minimal, right? They can be very prominent in the soft tissue integument, like the coloration of feathers, but not as obvious in the skeleton. And then with many archosaurs, the skeleton's sexual dimorphism might not be so obvious. And that's one of the problems, is that what we have only of fossils usually is the bone, scales, shells, and teeth, the hard parts. So it can be really hard to detect sexual dimorphism in the fossil record. So let's look at a few examples of variation in Fona. Let's go back to the jugal bone. This is a really important one. If we zoom in up here on the lateral surface, right, we can see that on that there's differences specifically in the level of rugosity and ornamentation between these different individuals. The three on the periphery have extensive amount of rugosity and maybe even a ventral ridge, things like that, while the two in the middle show smoother surfaces across this shield region, right? Variation also exists in a number of neurovascular foramen across skull bones, right? Here is a syringular of two Phona individuals, one of which is the left counterpart to the other. This bone here, the syringular, serves as the hinge joint of the jaw. And right around that hinge joint, right here, you can see that there are a number of neurovascular foramen. And if we zoom in on them and look inside of them with CT scanning powers, we can see that some foramina are consistent across the board, specifically the blue one, the red one, and maybe the green one. Whereas the other two, the yellow and the purple, vary in their presence and position. Variation also exists in other cranial elements, right? Here's the prefrontal bone, which is at the top of the skull. And in the Hall type of Phona on the top, we can see that this region possesses about 5 foramina, whereas in the paratype locality, mini troll specimen, there's only 1. So why is this important? Why is documenting variation important? Well, if we go back to how phylogenetic analyses are constructed, right, they're used to construct ancestral relationships. The inclusion of things like variable characters, which are not evolutionarily relevant, can produce spurious associations between taxa and clades, resulting in things like collapsed trees or wildcard taxa, which causes the analysis to be unable to confidently place certain taxa in the correct location. This is just one example of why variation is important. All right, I want to share with you now another cool example of variation in Phona, right? And so what we're looking at here on the left are 3 femora. The one with the red star is from one locality. The 2 on the right with the blue star are from another locality. And we can see that all the femora are about the same size in length, right? They're about like 20 centimeters in length. But when we look at the humerus, which is the bone that attaches to your bicep, the same specimen for the humerus in the Manolo locality, which is the one with the red star, is approximately 20% longer proportionally than the others, right? So we're getting scalar differences in some of these limb bones. And this is a similar pattern that we see in other limb bones, such as the bottom wrist bone, the ulna, right? Where it's also about 20% longer in the Manolo locality. And we also see similar patterns in the tibia and fibula. And then there's something really weird when we look at the pelvis. This is like an epicenter of variation in these animals. Right now, we're looking at a pretty normal pelvis. This is the pelvis from the paratype locality, the mini troll individual, right? And we can see that all the bones, right, the ischia and the pubis, right, ischia on the left in orange, pubis on the right in green, but they're all individually isolated individual bones, left and right counterparts. But when we look at the karmic Phona from the Hall type locality, the pubis is fused right down the middle of the midshaft. They're fused together. What is going on? And that's not even the weirdest part. If we look at the top left, right, we're seeing the obturator process. This is the process that's well known across dinosaurs, right? But in this individual of Phona, there's this massive perforation right through the obturator process. And that's not even the weirdest thing. Above that, right there, this little mount is a completely new, totally like unique accessory process. What is going on with this animal? So altogether, I concatenated and collected all of these different types of variations, ascribing etiology to each one of them as to whether they're the result of ontogeny, or could they be taphonomically variable, or is it just phenotypic variation? And so for the majority of significantly variable characters, I was able to rule out taphonomy, right? We can also eliminate interspecific variation because we've already established in chapter 1 that Phona likely represents one species. So that leaves us with sexual dimorphism and ontogenetic variation. So let's start with ontogeny. This is a variation due to individuals with different ages. But to explore this, we're actually going to take a pause from chapter 2 and pivot to chapter 3. Then we'll come back to chapter 2. So in order to estimate the age of fossil animals, we have to do something called histology, which is kind of like counting the tree rings to estimate years of growth. So I took 5 limb elements from Fona and performed this histological sectioning. The 2 with the stars above them are from the same individual from the paratype locality. We can see them here in the Corey map, preserved right next to each other. And then I sliced up these bones to make thin sections that are paper thin. This is an example of a turtle shell. This is not Fona, but this is just an example of what that process looks like. And then with the power of polarized microscopes, we were able to investigate all the bone tissue. So here's the smallest femur of Fona that is known from the mini-troll locality, and it shows no cyclical growth marks, and it bears bone tissue that's indicative of rapid growth, indicating an individual that is probably under a year of age. Here is the femur from the last chance therodromine Corey, with lots of growth marks throughout the cortex. And the growth marks, specifically what I'm talking about, are those red and green arrows in the window to the right. There are those red and green lines. These are growth lines that run circumferentially across the cortex. This is the femur from the mini-troll locality, also showing a few growth marks, although this is a distal section where it's known that fewer growth marks are generally preserved. But here is its counterpart, tibia, right? And here is the tibia from the holotype locality. So initially, I counted up all these cyclical growth lines, right? And I plotted them out here with the y-axis representing the distances between each CGM, cyclical growth mark, and the x-axis representing the number of CGMs from the endosteal surface, kind of representing time. However, we can't use growth lines alone because speedos are weird in that they're often shown to not preserve lines of arrested growth well. And even when they do, many of these can be ambiguous. So they do things like merging together, they fade away across the circumference of the cortex, or they can bunch up together within a single cycle of growth as multi-lag sequences. Therefore, with speedos, we have to rely more on assessing the changing types of zonal bone tissue to get our age estimations, relative age estimations. So here's the best example from Fona, right? We can see here that there are diffuse stripes running circumferentially, and the dark zones are areas that represent periods of rapid growth in which bone tissue was deposited hastily and haphazardly, causing the collagen fibers to orient in a chaotic orientation, which blocks polarized light. Whereas in the brighter bands, these are periods that represent slower periods of growth where bone was deposited more steadily and organized, allowing for more light to pass through. Altogether, I was able to determine that the individuals of Fona, which are the largest individuals of Fona, which are the ones that preserve the majority of skeletal remains, represent individuals at a minimum of age 4 to maybe 6 years old. Therefore, they're all skeletally immature subadults around roughly the same age. Okay, so now if we go back to chapter 2, we can kind of rule out ontogenetic variation, right? Kinda, but there is some nuance to discuss there that we'll come back to later. But next, let's check out sexual dimorphism. One of the biggest revelations is that it seems like there might be 2 distinct morphotypes of Fona. At this point, it's impossible to ascribe any specific sex to either one of these morphs, but it seems like one is a gracile morphotype and another is robust. And a similar pattern has also been noted in Fona's closest relative, Eryctodromius. If we go back to our locality map, this is where things get really cool. We can see that the holotype karmic seems to represent the robust morphotype with shorter, stouter limbs and increased rugosity across many of the cranial elements. In contrast, the individual from the Manolo locality seems to preserve the gracile morphotype, with some limb elements, like we showed earlier, that are proportionally 20% longer than those from Karmic. And most interestingly is that the mini-troll locality seems to preserve both morphotypes, the gracile and the robust, alongside that juvenile under a year old. So maybe we got a family. All right. Again, why is documenting all this variation important? Well, in 2016, Griffin and Nesbitt were able to show that across archosaurs, the ancestral condition that was inherited from amniotes is a condition of high intraspecific morphological variation, with only a few theropods and birds exhibiting reduced variation. And now we can confidently say that we can also add speedos in to demonstrate that they also have high rates of variation, filling in the gaps for this critical family. But like I said earlier, we can't rule out ontogeny completely, because even though the subadults of Fona are about the same age, there's an interesting quirk among non-avian archosaurs in which the acquisition of features across ontogeny is not always linear, but can occur at different times for different individuals. For example, there might be one individual at 14 years old that can acquire a trait, but another may have acquired it 3 years ago, and another will attain it in 2. And because of all the preservational bias in the fossil record, we tend to find individuals at the midpoint of life. It's harder to find really, really old individuals and really young individuals. What this means is that if we could wish back all the specimens of Fona back to life and put them in a zoo and watch them continue living on them to their natural death, we might see a significant decrease in their morphological variation. Maybe. All right, last but not least is chapter 4, where we notice something quite odd that was growing inside Fona and other speedos. So there are a number of other dinosaurs that have been named from our lab from the Mustn't Touch It. Moros was the earliest definitive tyrannosaur and featured in one of the Jurassic Park movies, I think. And then Iani, which we named 2 years ago, was a robust, medium-sized herbivore with a transitional skeleton that was grading towards what would eventually become those massive-bodied hadrosaurs at the end of the Cretaceous. And when we excavate these localities, the skeletal elements are usually disarticulated, like in this quarry map, which is a really bad quarry map, but it's just an example. But they're usually spread over large areas, and they're just generally less complete. And this is probably because, if you remember, the Mustn't Touch It was a floodplain environment where decaying skeletons were probably exposed on the surface for prolonged periods of time and would have incurred varying degrees of environmental transport and biological scavenging. But this is the exact opposite at our Fona localities, right? Usually skeletons are almost entirely complete. Almost every bone is preserved, and they're jam-packed into these tight bundles, and they can show articulation sometimes even in their original death pose, like with the holotype locality here, where we can see, we're zooming in, we're going to highlight those bones here in a second. Boom. And we can see that what we're looking at right now is stratigraphic bottom, almost like we're under the ground looking up at its belly. And we see its arms splayed to the sides, and then its leg kind of yoga tucked up towards its hip. So the initial hypothesis with this unique preservation was that, hey, maybe these animals are burrowing under the ground. Maybe they're doing the hard work for us by burying themselves under the ground. When their burrow collapses, then they're preserved there, and then they're not going to incur all of those environmental effects. But this hypothesis didn't come out of anywhere, right? Because if we go back to our friend from the YN Vaughan assemblage, Eryctodromeus was initially discovered and described in what was interpreted as a subterranean burrow. But unfortunately, an obvious burrow structure has not been found in the surrounding matrix around any of our Fona localities, and we don't exactly know why that is, but it might be because of some of the geochemical and sedimentological differences between the two formations. But needless to say, that's kind of where this hypothesis stopped for a while until I did histology. And then we noticed something quite odd. And it's this strange stuff here in the adult leg bone, the subadult leg bones. And it's this stuff called CCCB, which stands for compacted coarse cancellous bone, which is a bone texture known to occur extensively in many modern burrowing animals. You can usually identify it by its chaotic structure that looks very swirly and warped in cross-polarized light. And it's thought that CCCB provides higher resistances to the stresses and strains and torsional forces that are associated with repetitive biomechanical functions. And indeed, it's a similar pattern in tissue we see in the femora of Fona. And it's especially prolific in distal sections. It's quite extensive. So I wanted to see if CCCB existed in other ornithischians to get an idea of how prevalent this bone tissue might be across the whole group, and also see if we can pinpoint maybe clades or species that might be fossorial. So I scoured the literature for every example of every single ornithischian that has ever received histological sectioning, and I scored whether or not the authors documented or figured CCCB in the limb bones and scapulae. I also evaluated CCCB in these animals across a variety of criteria, such as the presence in what element, ambiguity in its identification, the prevalence across the cortex, the position in the section along the shaft, and the age of the individuals that were sectioned, and also the largest known body size for a taxon. And I took this data and mapped it out across the phylogeny. So on the right here, you can see if the color of the dinosaur's name is blue or light blue, it means it has CCCB or possibly has CCCB, and those in gray and black, it is absent or was never mentioned or figured. Although there doesn't seem to be any phylogenetic signal to the distribution of CCCB, there does seem to be a size bias. In this plot, all the ornithischians are plotted smallest to largest from left to right, with the femur length used as a proxy for body size. And what seems to be the case is that CCCB is prevalent among ornithischians with femur lengths that are below 50 centimeters and nearly completely absent in those above 50 centimeters. But in order to evaluate the confidence of this relationship, I ran something called a phylogenetic ANOVA, which allows one to test the statistical confidence between a discrete variable, in this case, the presence or absence of CCCB, and a continuous variable, in this case, femur length, while also considering the potential influence or biases that are imparted on by evolutionary relationships, which is the phylogenetic part of the ANOVA. And from this, I was able to recover p-values well below the 0.05 threshold, suggesting that even when accounting for phylogeny, there is a strong statistical support for the relationship between body size and the presence or absence of CCCB. So is this a nail in the coffin on the burrowing debate? Since Fona and all these speedos have CCCB, they must be burrowing, right? Well, not so fast once again, because CCCB is also found in many other taxa, such as turtles, pterosaurs, and even some large graviportal creatures. It's strongly influenced by ontogeny, being more common in juveniles and generally decreases in abundance towards maturity. So it could just be that Fona and other speedos, it's simply related to any repetitive biomechanical motion that they're doing, which could be burrowing behavior, but it could also include things like running and foraging. And lastly, it could also just mean that CCCB is frequently absent in large body taxa, simply because the residual tissue from early ontogeny gets completely obliterated during the extensive remodeling that's necessary to grow a small bone into such a large bone. But ultimately, the presence of CCCB is not a direct indication of fossoriality or any specific biomechanical behavior, but if it is absent, it probably means that these animals are not burrowing. And with that concludes my defense. Okay. I have an insurmountable number of people to thank today. A humongous number of people have been in along this ride with me, going all the way back to high school, my peers, my friends, my family, my colleagues. See if you can find your face spinning. Thank you so much to everyone. That's the quagmire of friendship, by the way. And then here's my official thank you and acknowledgements. So many people to thank. First and foremost, I would like to thank my advisor, Lindsay. Right, she has been, what can I even begin to say? She has made my dreams come true. And she saw the potential in me before I could even see it in myself. And then there's a long list of people to thank. My committee members, right? The lab PIs that have been my mentors and teachers, all the preparators that worked on Fona to get it prepared out of the rock and into a state that it can be studied. All the lab postdocs and researchers that have been through the rotating doors of our lab. My lab trivia crew, right, all around here. You guys made the hard days better and the good days funner. My research students that have stayed curious, and especially to the Department of Biological Sciences, specifically Freya, which has been like super helpful for figuring out all this stuff. The many museums that I visited during my my travels, specifically the Museum of the Rockies, which I took more trips to than any other institution. My Gurkha Squad bros in the back there, and my new in-law family that's online, I think, right now, and then my Reinerside family, my Chamorro ancestors, my close family, and then my bride-to-be very soon, Emerald Bender. So thank you again, and with that, I'm happy to take any questions. All right, let me pull up the Zoom stuff too, just to see. Oh, sweet. Howdy, folks online. Right on. Yo. Oh, thanks so much, guys, online. Right on. You guys rock. Okay. So anybody from the audience have a question? Because there is one from thanks, Aurora. Right on. There's one from Kai. Oh, okay.
Awesome
Let's
job,
go
Thank you
do
so much.
first, and then we'll get Kai as he texts it. He'll send it.
I wanted to ask if there was an uptick in the abundance of secondary osteons in the elements or the species in which you saw the compact trabecular bone. In the sense that we've observed in like sauropods, things that have a lot of weight bearing, of like a huge increase in secondary osteon formation in the cortex of the humerus and femora. So I was wondering if there would be an association with that remodeling in animals that are also doing a lot of, if it's needed more compact bone, like you can see more remodeling, possibly for like microfracture repair.
Yeah.
In those same elements.
Yeah. So there definitely is an association with secondary osteons where CCCB is present. I've also seen an extensive amount of secondary osteon, but the specimens that I sectioned of Fona, which is just the femur and the tibia, there's never like the haversion tissue. There's never like an extensive amount of secondary osteons. It's mostly primary tissue, but there are specific places like the anterior lateral plug, right, in the tibial section, which is kind of like a muscle attachment joint area that does have, you can see a ton of like secondary osteons forming in those areas. It's also pretty prevalent more so towards the endosteal surface. So.
I would like wildly speculate. I wonder if that's associated with areas that are more likely to minor stress fractures or something like. I don't know.
That's a good
this
point.
Is totally just a wild kind of.
No, that's great. I agree. I think they probably go in tandem to providing like this different mechanisms to help the same biomechanical stresses. Yeah.
Because if there is a cord, that would totally make sense to me.
Yeah.
Like, yeah.
Yeah, cool.
Also very good time.
All right, thank God. Thank you so much. No, right on. Cool. Okay. While you guys prepare your questions, if you have any, let's take one from Kai online. Oh, what's up, Josh? Hey. Whoa, what's up, bro? Did you see your head spinning? Okay, you were in the last slide. So Kai says, it looks like your work doesn't fully show that Fona was fossorial. What other data would you need to say it with certainty? Well, a burrow would help. I think that would be a cool place in the future for somebody to study, is looking at maybe there are some signals in the grain orientation of the localities. Maybe if we did some sort of micro survey of a section of the Corey map, we might be able to detect something like that. More sampling, more specimens of Fona would help. But I think at this point we would just really need to find a burrow. So right on, Kai. Thanks. He's coming out to help us prospect. He's gonna find one for us. Right on. Okay. Any other questions, my friends? Yes, Christian.
Yeah, I mean, what it would be if these animals are burrowing, let's say Eryctodromeus at least, what's the inferred burrowing mechanism? I mean, in most extant burrowers that you've compared them with histologically, these are like mammals that are powerful forelimb-based burrowers.
Yeah, for sure.
Dinosaurs don't have the same degree of forelimb.
Exactly.
So how do you hypothesize these things would have been burrowing?
I've thought about this for a long time, and I agree with you. So in retrospect, I wish we had sectioned a few of the forelimb bones, like the humerus. That might be a place to go in the future, but it's almost like certain that they're not doing the majority of the burrowing with their hands. Especially when we look in modern bipedal fossorial animals, things like Jerboa, things like there's another bipedal mammal from like a desert. I can't remember what it's called right now. It's the spring hare, yes. And then there are things like bush turkeys in Indonesia, which are true like bipeds. They don't even have forelimbs to help burrow with. But the Jerboa specifically and the spring hare, what they're doing is they spend a lot of time using their hands to scratch away sediment from a wall, right? Or from the walls of the burrow that they're constructing. And then they use their powerful feet to just like launch that stuff backwards. They're really using their feet a lot. The same is the case with bush turkeys, which, you know, they're not really like digging underground, but they are digging in the sense that they're creating like extensive nests with tunnels, and they're using their feet entirely. It's those kicking motions, those backward kicking motions. So I imagine that Fona and Eryctodromeus are probably mostly utilizing their I mean, this is all speculation at this point, but I imagine that what they might do is use their hands for stability in a tunnel, right? Or removing the sediment from the sides, and then they would be kicking out the majority of that sediment with their feet. And there's also potential that they could be using their face a bit. So I think that's an interesting area to look into, but it could be that they're using their rhamphotheca and maybe some of their premaxillary teeth. You know, I've thought about this a lot actually, because what we noticed with my student Adam, I think Adam is online right now, but we did a SVP poster a year ago, 2 years ago, where we've noticed an incredible overabundance of premaxillary teeth relative to cheek teeth, right? So it seems like the Fona is shedding their premaxillary teeth at a ridiculously higher rate than their cheek teeth. And like there's a Joholosaurus paper that came out that showed that, yeah, premaxillary teeth do seem to show like faster replacement rates, slightly faster replacement rates. But I think the numbers we got was like an order of magnitude. It was like huge, like vastly higher. So it could be that they're just wearing down their premaxillary teeth at a higher rate. And there does seem to be interesting weathering patterns on some of the teeth, but at this point, it's kind of just still speculative. So, yeah. So many premaxillary teeth, Adam says. Right on, Adam.
Yeah, I think we have time for one more question, and then we have to call it last chance to grill with you. There we go.
Bucky.
Oh, sorry, go ahead.
Go ahead, Bucky.
Okay, so you found two different shape morphs, gracile and robust. And so when you look at modern animals, the ones that are monogamous tend to be much more similar in shape and size. But you're finding these, it seems like in burrows, right? You have one that seems like it has a family. So were they monogamous?
Oh, that's a good question. I mean, maybe that was a very naughty burrow.